So from here, we collect our data from all of our participants on if they caught a cold or not, and how severe and how long their symptoms lasted. And then we analyze that data using statistics. And those statistics help us look for patterns and relationships in the data that we've gathered, which allow us to draw conclusions. And this helps us to see if the findings that we draw from our data support the hypothesis that we started with. If our experiment shows us some results that are not what we expected, that don't support our initial hypothesis, that doesn't mean we're wrong. It's not a bad thing. Most scientific experiments end up not supporting the original hypothesis that we started with. This just means we gotta go back to the drawing board and rethink our hypothesis and try again, or try something different. Okay, so this is from a slightly different study, but it's still a study looking at the effect of echinacea on the duration of a cold. And often results of an experiment are displayed graphically in order to help visualize the conclusions that can be drawn from that data. So here we have our independent variable, echinacea versus placebo. So the two things that we changed are these two lines. Okay, so placebo is here, echinacea is here. And then our dependent variable is the total symptoms, how intense their cold symptoms were over the extent of the study. So placebo, echinacea, it looks like echinacea they have slightly better cold symptoms, but because these data points are overlapping, see these bars right here? They're overlapping between these two groups. That means there's no statistically significant difference. We cannot say, based on the sample size that is here, based on the type of data that was collected, that there is a statistically significant difference in the symptom score for the echinacea group versus the placebo group. So this data graphically is showing us that there is no difference in symptoms and cold symptoms when taking echinacea versus taking a placebo. So this was published in 2020. This specific study that we're talking about also showed the same thing. There was no difference in either having cold symptoms or the duration of the cold between any of the four groups. Those that took echinacea before exposure to the cold versus those that took echinacea after exposure to the cold virus. All four groups were just as likely to catch a cold and have symptoms that lasted at least 3 days. So we could say, okay, maybe that's what this specific study found. There's no difference in catching a cold or the duration of the symptoms, but maybe that dosage of echinacea wasn't high enough. Or maybe there is an active ingredient in echinacea, and maybe if we were to increase the concentration of that active ingredient in echinacea, that could lead to decreasing the duration of a cold, right? So this isn't saying stop studying echinacea. What this is saying is that in this specific setup, we did not see any difference between echinacea and placebo on the effects of cold symptoms. So there could be something here that this specific experiment did not let us look at or did not explore in depth. Okay, so how do we get from this to publication? How do we get from experiment to running analyses of our data to making conclusions to sharing out those conclusions? So science is communicated by researchers in what we call primary literature journals. And in order for research to be published in one of these primary literature journals, research has to go through a really intense and rigorous peer review process. And this peer review process is done by other experts in that field. What happens is they read through the experiment, they read through the data, they read through all of the methods, and they pick it apart, and they try and find areas that need improvement or provide guidance on a different way to approach the experiment or the methods or the data analysis. So less than 1/5 of all papers that are submitted to a journal are ever published at all, are ever sent out to the wider scientific community. Most often, the papers that are rejected from the journal are rejected because their methods are not a suitable way to explore that research question. So the experiment that they used was not a truly good fit for exploring their research question. Or their data was not robust enough to support their conclusions, meaning maybe their sample size was too small, the signal that they found in their data, any sort of patterns or relationships that they found in their data, was not significant enough to actually support those conclusions that they were claiming. So this is a very lengthy and intense process that happens after years of running experiments that gets written up into a scientific journal, which then gets submitted, gets sent back for edits, it's revised, hopefully it's accepted, and then it's published. From there, the papers that are published are sometimes picked up by the media, think like newspapers or news outlets, things like that, and that can sometimes change the narrative or misconstrue the ideas that were being communicated in that paper, mostly because science can often change. The more data we collect, the more experiments we run, the more precise we are in our understanding of how something functions. But all science is evidence-driven, empirical data that goes through thorough analysis, which draws us to very careful conclusions. And when new data comes out, that new data, that new information might conflict with what was found previously, because now we just have more information now, we have more data. We know more about how this works. And so we can make a more reliable conclusion with additional data. And this has been particularly salient with the coronavirus pandemic, right? So at the very beginning of the pandemic, some people said, don't wear a mask. That might actually increase the risk of catching the coronavirus. Then later on, it was suggested that we should wear masks, right? Because after we were able to spend more time with this virus, get to know it better, learn more about it, and learn more about how it spreads, that led us to new data, which led us to different conclusions. Okay. So new data might mean that our old previous conclusions are reevaluated and revised, or even discarded entirely. So the more we know, the better conclusions we can draw. Okay, so science is not static, it could change, right? Like this example, linking autism and vaccines. There was a paper published in 1998 in the British medical journal called The Lancet that drew a connection, a cause and effect connection between vaccines and autism. That paper resulted in many scientists also trying to investigate this idea. So this paper was published in 1998. After that, hundreds and hundreds of studies tried to research and investigate this idea that there's a connection between autism and vaccines. Not a single study that was published after this 1998 paper in The Lancet was published could confirm reliably that connection between autism and vaccines. So, because more data was published, none of that data supported the original conclusion of this 1998 paper. That 1998 paper was what we would say retracted, meaning it was later determined to be found false or not true after we learned more. So Andrew Wakefield, who's the author of that paper, actually was stripped of his degrees. He had some honorariums also that he was stripped of because it was later found out that he manipulated some of the data. His subject size, his sample size was very, very small, and he did not follow good scientific processes, which, even though it may have been a little bit late, the scientific community and that process of peer review eventually caught. Oftentimes, when we see science in popular media, we often see a headline. We might read the first couple paragraphs of an article, we might even read the entire article itself, but how often do we actually go back and read the original paper, the original publication that the scientists wrote that articles are based off of, right? So for example, I just did a quick search on red wine healthy on Google News, and I came up with these two articles were popped up. One was published a week ago, one was published 2 weeks ago. One claiming that alcohol can cause atrial fibrillation, which is a heart condition that can be very, very bad. The other one saying, no, red wine can lead to lower blood pressure. Okay, so it's never ever as simple as the headlines make it out to be. In this study, they actually used 100 people that had a history of atrial fibrillation and tracked them for 4 weeks, monitoring their alcohol intake and cardiac rhythms. So these people already had a propensity for atrial fibrillation. This study right here was looking at participants' food intake, their gut microbiome, and their blood pressure levels. And what this study found is not necessarily just red wine, but it's any sort of flavonoid-rich foods that could lead to potentially lower blood pressure. And they didn't run an experiment. They did this as a questionnaire. So they asked people, how many times did you eat these certain foods, right? There was no controlled experiment where they said, you will eat this, you will eat that, you will not eat this, you will not eat that. Okay, so it's never as simple as the headlines make it out to be. And even the articles themselves often simplify the experimental conclusions and take away the complexity of the results that they found. So there are limits to science. Science is an empirical process, meaning that we use data to make conclusions, and we can't collect data about things that we can't measure, right? So science can only answer questions or help us to answer questions based on things in the natural world, things that we can measure. Other things like moral statements or value judgments, those are subjective, can't measure whether or not something is good or bad per se. Anything that's subjective falls outside the realm of science. Anything that is based in the supernatural realm falls outside of the realm of science, right? We can't ask questions about whether there's an afterlife or whether there's a God. That is outside of the realm of questions that science can answer. And then lastly, we have technology, which is basically just the application of scientific research. Lastly, just want to touch on this idea of a hypothesis versus a theory. So there's sometimes some confusion about what this word theory versus hypothesis means, because in our everyday language, theory often is used to talk about a hunch or a guess or a speculation that we have, meaning just generally something that we're not certain about. We, as scientists, kind of use those terms in opposite meanings. A hypothesis is something that we're not certain on, versus a theory is something that has a lot of support and evidence behind it. Right, so again, a hypothesis is just a proposed explanation for a phenomenon that has been observed, versus a scientific theory is an explanatory hypothesis for natural phenomenon that is well supported by lots of different types of empirical data. A scientific theory is often a general statement about how the world functions or how the universe functions, right? So it's a hypothesis that's a general hypothesis that has withstood the test of time and has a wide foundation of empirical evidence that has time and time again supported that theory, that hypothesis. If new evidence comes about that runs counter to that theory, then that could trigger the process of rethinking and reevaluating that hypothesis that the theory is drawing from. So just because something is a theory now doesn't mean it will always be a theory forever. Science is flexible and incorporates new data and new information as it comes about. So some theories that we'll talk about in our class are the theory of evolution and natural selection, and the cell theory, right? And in biology, oftentimes, we don't have laws, but we have theories. And theories are broad scope hypotheses that have a large amount of empirical evidence supporting them.